Alrighty, good morning. Going to talk a little bit about, we're going to pick up photosynthesis where we left off. Um, so we last talked about the light-dependent reactions with photosystem 2 and photosystem 1. So now we're going to move on to the light-independent reactions, which is really just one step, and that's called the Calvin cycle. So, kind of like with the last time, I'm going to introduce this first, and then I'll explain the crazy diagrams for you. So after photosystem 2 and photosystem 1, the next step is the Calvin cycle. So this was a guy named Melvin Calvin. Kind of an interesting name there, huh? But he was the guy who figured out how plants made the organic parts, so the sugar or the glucose. So since he was the one to figure that out, this whole step is named after him. So that's why it's called the Calvin cycle. So just a random tidbit of information in case you're ever on Jeopardy and they want to know who was named after you, would say Melvin Calvin. So, all right, so with your Calvin cycle, this is going to take place in the stroma of your chloroplast, that semi-fluid matrix. And what's going to happen is you're going to take that energy that was produced in photosystem 2 and 1, and you're going to take that coenzyme NADP plus, and you're going to use those and fixate your carbon into a glucose molecule. So you're going to take that, you're going to pop those carbons from the carbon dioxide, and then you're going to take those carbons and basically piece them together, or fixate them together to make those glucose molecules. So this is sometimes also called C3 photosynthesis. So any plants that are considered C3 are going to go through those, and that's going to be a lot of common plants. So like beans, carrots, spinach, a lot of trees. So, okay. So now go ahead and look at your crazy diagrams here on the Calvin cycle. Again, it's in the stroma. I'm going to start on the left side. We're going to start over here with this RUBP molecule, and we'll cycle through, and eventually we'll end back up here with that 5-carbon molecule. So I drew it out. It's called RUBP. And what ends up happening is this molecule is worked on by Rubisco, by an enzyme Rubisco. And what happens is the carbon dioxide from the air that's taken in through the guard cells in your leaves, it's going to detach that carbon from that carbon dioxide that's been taken in, and it's going to fixate it onto that 5-carbon RUBP molecule to form a molecule that is called RUBP carboxylase. So there's another enzyme called oxygenase that's going to hold that carbon dioxide together as it attaches to the RUBP molecule. So then what's going to happen is that 6-carbon molecule is going to split into 2. And we're going to call these PGA, phosphoglycerate molecules. And so there are 3 carbons. So basically, I'm only going to describe the outer ring there, the outer track, but it's the same thing's happening on that inner track. So everything's happening twice, essentially. So we're going to follow the top phosphoglycerate molecule. ATP from photosystem 2 and 1 is going to come in, and it's going to rearrange that molecule a little bit, and in so doing, it's going to lose that phosphate group, and it's going to become ADP, and it's going to transform that phosphoglycerate molecule into another 3-carbon molecule called a 1,3-bisphosphoglycerate, a 1,3-bisphosphoglycerate molecule. And again, that's happening on the inside circle there too. Then we're going to take that NADPH that we formed all the way back in photosystem 2, and then that's going to come in and rework that molecule, the 1,3-bisphosphoglycerate, and it's going to create another 3-carbon molecule called PGAL. Then what's going to happen is these PGAL molecules are going to recombine into a 6-carbon sugar. One of those sugars is going to be popped off, and it's going to kind of hang out. And the remaining 5 carbons are going to go back up, and you're basically left with RUBP again. What that 1 carbon down there is going to do is just kind of hang out until you get, and the whole cycle is going to go through again. And when it reaches after that PGAL combines again, it's going to pop off another carbon. Okay, and what's going to happen is that new carbon is going to join to that one that's been sitting there. So now you've got 2 carbons down on the bottom. And we're going to go take in some more carbon dioxide and go through the cycle again. So we're going to go through the cycle 6 times until we get that 6-carbon molecule of glucose. And that's how you're going to produce the glucose molecules. So we figured out yesterday about the how you get the oxygen molecules from the photolysis. And then we figured out now how we're popping off that off of the carbon dioxide up there at the top of the Calvin cycle, and so you're left with oxygen that we're going to breathe into. But you're also that oxygen will also be added to that glucose molecule, and then now we're figuring out how you're getting the glucose molecules as well. So this is, like I said, called the C3 photosynthesis. Now there are a couple of other versions of photosynthesis. The next one is the C4 photosynthesis. So plants that are considered C4 plants are going to go through this. C4 plants are thought to evolve after your C3 plants in tropic areas that have high temperatures and really dry conditions. So they can outcompete those C3 plants in dry conditions. Now, that's not going to be the same as the CAM plants that we're going to talk about at the very, very end that are in really, really dry places. But some examples of some C4 plants, some of your grasses, like your crabgrass, is going to be a C4 plant. Corn is a C4 plant. So, so there's a couple ones there. So they're able to kind of monitor their water and things, but not like I said, not as much as the CAM. So a guy named Hugo Kortschak figured this out by studying the sugarcane plant, and so he was the one to really figure out that there was this other step that was occurring with certain plants in these C4 plants before they would go through the Calvin cycle. There was kind of an intermediate stage there, and then Slack and Hatch were the next 2 people to confirm that there was indeed this secondary process that would occur in the mesophyll. So you're going to have, your C4 plants are going to have basically two different steps. You're going to have an intermediate step here in the middle in the mesophyll cell, in the stroma of the mesophyll. And in this case, I've written this down here for you, but we've got a PEP molecule, and it's going to go through a couple of processes before it gets into the Calvin cycle there. So I'll explain the rest of these things like the PEP carboxylase and the oxaloacetic acid, things like that, but those are right there for you. And then the rest of the Calvin cycle is going to occur in the bundle sheath cell. So, alrighty, so I'm going to draw your attention to the pictures first here. So, with your C4 plants, you were first starting out in the mesophyll chloroplasts, again, still in the stroma. And again, we're going to start on the left. We've got a 3-carbon sugar, or a 3-carbon molecule here that we're going to call the PEP molecule. And what happens, it's going to be really similar to that, to the Calvin cycle that's occurring here next door. And you're going to have an enzyme come in, PEP carboxylase, and it's going to take that carbon from the carbon dioxide, and it's going to fix that onto that PEP molecule. So then what's going to happen is now we've got a 4-carbon molecule called oxaloacetic acid. That gets transformed into either malic acid or aspartic acid, and those will leave the mesophyll cell through the plasmodesmata and go into a bundle sheath cell, specifically into one of the chloroplasts of your neighboring bundle sheath cells. In which case, one of those carbons from that malic acid or the aspartic acid is then going to pop off and join the oxygen, and then that's going to then enter into the Calvin cycle. And then the rest of that Calvin cycle should look familiar. Okay? The remaining, after we pop off that 1 carbon, we're left with a 3-carbon pyruvic acid. So it's the PEP molecule, the phosphoenolpyruvate acid. And so then that will go back into the plasmodesmata and go back across into a mesophyll plant, mesophyll chloroplast, and then we'll start the whole process all over again. So it's called a C4 plant because you've got the 4 carbons there initially instead of the 3 or the 5. So, all right, last part, CAM photosynthesis. Your CAM photosynthesis plants, they only occur in a very specific family of plants, the Crassulaceae. Basically, your pineapple and your cactus plants. So, with these guys, very much desert plants, desert type plants. And so, the reason why these are even more so different than the other 2 types of photosynthesis is because part of this photosynthesis actually occurs at night. So it doesn't require the light because it's a light dependent reaction, and so some parts of it are going to occur in the evening when there's no light, when the guard cells can open to minimize that water vapor loss. And then it's just going to kind of chill until the daylight, and then it will finish going through the photosynthesis with sunlight. So that's what makes these CAM plants different. So it's called Crassulaceae, Crassulaceae, acid metabolism. So Crassulaceae acid metabolism. I have no idea if that's really how you pronounce the Crassulaceae. It's been like 11 years, so even more so than that, I guess. I've been out of college for 11 years, so it's really been more like 14 years since I I learned this, so it's been a while. So I'm not sure if I'm pronouncing it right, but okay. So to look at your diagram, it's your last diagram. You've got your CAM photosynthesis there. We're going to start on the far left with the 3-carbon PEP molecule. So with this one, it's going to look really familiar to the C4 plants, except this is all occurring at night. So all the stuff that kind of happens in the mesophyll chloroplast and the C4 plants is going to happen at night during CAM photosynthesis. So you got your 3-carbon PEP molecule. That PEP carboxylase is going to detach that carbon from the carbon dioxide, and it's getting that carbon dioxide because the guard cells are open since it's night. Okay, so which implies that the temperature's lower, so you're still going to have some water loss when you open those guard cells, but it's not going to be quite as much as if you had tried to open them in the middle of the day. So you're going to form that 4-carbon oxaloacetic acid. It's going to be transformed into that 4-carbon malic acid. And what's going to happen is your vacuole is then going to store this pool of malic acid. So we're not actually going to go on through the plasmodesmata into the Calvin cycle. We're going to wait until daylight comes, because we're going to store all this up. We're going to store up as much of the malic acid as we can, because we won't be able to open those guard cells until night again. So they want to maximize everything, and they need the ATP and the NADPH from photosystems 2 and 1, but that can only be made during the day. So that's why it's going to store that malic acid. So because it can't go through all of that, it could technically go through the Calvin cycle, but it's not going to have the tools it needs to finish it, because it won't have the ATP to split things. It's not going to have the NADPH to maneuver those carbons around. So when the sun comes out, the guard cells close, and then the malic acid goes back into the chloroplast, and then we go through your Calvin cycle. And we're able to also go through that photosystem 2 and 1 to produce those ATP and NADPH. So, but we have a finite supply of malic acid. Once that malic acid is used up, in theory, the plant would stop photosynthesizing, because it's not going to open those guard cells in the middle of the day. So that malic acid then is going to be changed back into that 3-carbon PEP like it was up in the C4 plants, and then it's going to go back in and then just kind of chill out and wait until the night, and then the guard cells will open, and it will start combining with this carbon dioxide to make that oxaloacetic acid again. So hopefully that makes sense. I hope it does. And so I know that was probably a lot. It's way more intense than I think people realize. On the back side of your things, I went ahead and made a chart for you with C4 plants, C3 plants, and the CAM photosynthesis. Which ones go through light dependent reactions, which ones have extra steps, which ones go through the Calvin cycle. And really, everybody goes through the light dependent reactions, everybody goes through the Calvin cycle, and then the extra steps are just going to be the C4 and the CAM. But hopefully this makes some sense for you. We'll probably have a quiz over this stuff sometime next week. I'll probably go ahead and have you take a quiz over that at some point next week, probably Wednesday, because I hate to do that to you on Tuesday since I know you're gone Monday. But I don't necessarily want to make it Thursday either, unless you're okay with it. So I'll plan on a Wednesday quiz for you. If you'd rather have it Thursday before we leave for spring break, just let me know. So if you have any questions, just feel free to send me an email, and I will be more than happy to answer. So have a good day.